We're going to finish up the first part of this DNA section by talking about something called DNA replication, which is an extremely important part of DNA, and it's also where a lot of things go wrong, actually. It's where a lot of mutations occur and things happen where somebody might develop some kind of a genetic mutation, like, for example, cancer. So let's talk a little bit about why DNA replication happens, and then we'll talk about how it happens. So first off, let me talk about why it happens. Every time a cell divides, the first thing that needs to happen before that cell can divide is we have to replicate that DNA. Why? Why would we need to do that? Well, think of it this way. Your DNA is a cookbook, and I'm going to refer to it as that often. And that cookbook is a recipe book for every single protein that gets made in an organism. So what are some examples of proteins that get made in organisms? Well, we talked about these already, but muscle, bone, hair, skin, enzymes, hormones, neurotransmitters, all these things, pretty much every single thing that is in a living organism is a protein. And all those proteins have recipes. And how does the cell know which protein to make? That's held in the DNA. So the DNA is going to be in here, inside the nucleus. So let's say I want to make 2 new cells. I want to have the cell divide, right? I'm growing, or I hurt myself, or, cells divide all the time. Right now in your body, millions of cells are dividing all over the place. So when that cell divides, before that cell can divide, the first thing that has to happen is we have to copy that DNA. Because when we have 2 new cells, it is extremely important that those 2 new cells have their own copy of their DNA, because those cells need to be able to survive, function, and make proteins. So the first thing that has to happen is this DNA has to replicate. It multiplies by 2. And that happens in a very, very specific manner, and it happens very quickly, and it happens the same way every time. And that's what we're going to go over today. How does the cell actually replicate its DNA? So basically what it says here is every time a cell wants to divide, the DNA has to copy first. So it each has its own genetic copy. Like I just said, it would be like if I had a cookbook with every recipe that ever existed, and I'm the only one that has this cookbook. If I want you to be able to make recipes, you need a copy of that cookbook, right? So if you leave and never come back and I don't see you again, I need to copy that cookbook for you. And that's what happens here. The DNA gets copied, and the entire strand gets copied. So there's a lot of base pairs in a strand of DNA. So let's talk about how that happens. There's actually 4 major steps, and I'm going to just run through them with you, and then I'll show them. I'll show you some animations that'll hopefully help you to understand it. The first step is there's an enzyme. Just like everything, there's enzymes. And here's a little thing that's kind of funny about this. An enzyme is a protein. So an enzyme is also coded for by DNA. So if there's no DNA, there's no helicase. And if there's no helicase, there's no way to copy DNA. So every single component of this process needs DNA. So the DNA is first unzipped by an enzyme called helicase. Now, one thing I hope you remember about DNA is that the 2 separate strands, there's 2 strands that are attached to each other by hydrogen bonds. And I told you the thing that makes hydrogen bonds special is that they're a weak bond. And helicase is able to very easily unwind those strands. So if you take a look over here, you can see this would be a strand of DNA, and at this point the helicase would unwind that DNA strand. It's the same thing as if I want to read a recipe. The first thing I need to do is I want to open up the book. If I want to copy that recipe, the first thing I need to do is open that book, and that's what helicase does. Helicase cuts the and unwinds the DNA strand and essentially unzips it, and it breaks those hydrogen bonds. So that's the first thing that happens. Those hydrogen bonds get broken. After that, we have the unwound strand acts as something called a template. A template is just something that you use as a way to know what to do. So for example, a template in the case of building something would be an example. So for example, if I want to build a model, I would have a template to look at so I know what the model is supposed to look like. That is what the DNA strand acts as. It acts as a template to produce 2 new strands of DNA. So essentially, the third step is another enzyme comes along. So there's 2 enzymes you need to know for this process. The first one is called helicase, and that unwinds the DNA strand. Essentially, it opens it up so that way it can be copied. And then the second enzyme is called DNA polymerase. And DNA polymerase reads the strand. It attaches to the DNA strand, and it adds a new base to the appropriate spot for every nucleotide that's there. So it adds the complementary base. So if there's an A there, it adds a T. If there's a C there, it adds a G. And that process continues all the way down the strand until the entire strand is replicated. There's no 2 identical DNA molecules. They're always going to be a little bit different. but the strands, the two new strands, are going to be identical to each other. So my DNA is never going to be identical to your DNA. However, when my DNA replicates, there's the original copy, and then there's something called the complementary copy. And the complementary copy is the complementary strand, is the new strand that was copied from this process. So I'm just going to show it to you in this diagram here a little bit, just kind of review that with you so you know what's going on. All right, so what I have here is I have my DNA strand, right? And this shows you a DNA strand up here. And this is a DNA strand that I want to copy. So the first thing that has to happen is I have to unzip it, right? So the helicase is going to come along and it's going to attach to the strand. And it's going to attach to it at the beginning of the strand. This copies the entire strand, remember. You're not copying a section of the DNA, you're copying the entire thing. So when it attaches to it, it actually is going to unzip it. So bear with me as I draw this out here. It's going to take me a sec. So it unzips it. So let's say this is my strand of DNA here, right? It's going to unzip it and open it up. So you're going to end up with something that looks like this, let's say. And we'll put our helicase over here just to represent that that is attached at this point, and it's going to form the strand. So I'm going to just fill this in for. So my helicase has unzipped my strand, and I have my original. This is going to be my original strand here. So I'm going to put a little O here just to signify that it is the original strand, because that's what was opened by this. So this is our original strand. But now I need to copy this DNA, right? The whole point of this is to copy it. So the helicase unwinds it. So now we have our second enzyme here, our DNA polymerase, and that's going to attach to the strand, and it's essentially going to read it. And it's going to attach a new base pair to the complementary strand. So this acts as a template. So if I take a look here at my compound, I'm just going to move this back over. G goes with C. And then A goes with T. So basically, DNA polymerase is going to move down the strand, and it's going to attach a new base for each one that's there, the complementary base. So C goes with G, and A goes with T, right? So that's going to be our new strand. And then the same thing happens on this side. And it does happen a little differently on both sides. There's something called a leading strand and a lagging strand, which we're not going to really worry about for right now. But basically, what it does is it just forms two new strands, complementary bases for each one. And it's going to go through this process all the way down until it gets to the end. So it's going to copy the entire new strand. And what you're going to end up with is you're going to end up with a new strand, which I'll just make it purple here. And if you take a look on this side, I have a new strand over here. This is what's called the complementary strand. So we have our original strand and our complementary strand. Helps if I put a C the right direction. And one thing I want you to notice is if you take a look here, it goes G-A-A-C-T, C-T-T-G-A, and on this side it goes C-T-T-G-A, G-A-A-C-T. So these strands are identical to each other. There's the original strand and the complementary strand. And this process is going to go all the way down the strand of DNA until every single base has copied. Now, as you can imagine, are there sometimes mistakes? Yes, of course there's sometimes mistakes. Sometimes the wrong base attaches. Something copies wrong. There's 3 billion with a B, 3 billion base pairs. So imagine writing out 3 billion letters and copying each letter exactly. Are you going to make a mistake every once in a while? Yeah. Now, sometimes those mistakes are completely harmless, and sometimes those mistakes cause things like cancer or certain terrible genetic mutations. So this is exactly, though, how the process works. The replication occurs, and then at the end of the process, you end up with 2 strands of DNA, the original and the new strand, and it's going to be a complementary and the original strand. Thank you very much.